In this video we will discuss about this NK92 mediated anti-tumor mechanism. In the previous video we have understood that this NK92 cells which are genetically modified, and within that the CD-16-CAR-NK-92 cells which has this intercellular domain 41BB has shown more cell lysis compared to the other cases when they co-cultured with their respective target cells in the presence of the antibody, that is, in the presence of this antibody dependent cellular cytotoxicity. So the researchers wanted to find out the mechanisms. So what are the common mechanisms by which this immune cells target these cancer cells is by apoptosis. So they check the apoptosis in different co-culture conditions. So what are the co-culture conditions they use to check whether the apoptosis is the reason for the cell lysis? Is that they use the NK92 cells and its different genetically modified version, that is NK92 cells with CD16 cDNA and NK92 cells with CD16-CAR with intercellular domain, that is 41BB, and co-cultured them with HER2 positive cells and HER2 negative cells. So in terms of the HER2 positive cells, they used BT747 and MCF-7. The HER2 negative cells, those results are not shown here. For that they use MD 468 cells, which is also a cancer cells, but it is HER2 negative cells. So the first idea is to check the cell death using tryphan blue. So tryphan blue is the dye that can penetrate inside a dead cell membrane. So what they did is they co-cultured NK92 cells and their respective target cells in the presence of the antibody, that is Trastuzumab at 10 microgram per ml, and for 8 hours added two effector to target ratios, that is 4:1 and 8:1. Some of the conditions are already been established in the previous experimental results that we have discussed. So after 8 hours, what they did is they washed the effector cells, as the effector cells are the suspended cells and the target cells are the adherent cells. So it's easy to get rid of effector cells after this co-culture assay, and then they stain the this target cells with a tryphan blue. For the control we can see signals very clearly, but for this target cells the signals are very low. So the reasons that they have identified is that even that cells are very prone to remove to get rid of or to be removed from the cell surface through using in this 1x PBS washing step. So although they see some dead cell signals, but these results are not convincing. So what they did is they did move to the next assay or another assay, that is a CKK assay, CCK8 assay. In that case what they did is they use the flow cytometry to check the percentage lysis, and here they checked the apoptosis percentage lysis or the percentage lysis cells, that we can say that. So in that case they shown the results for the HER2 positive cells, that is MCF-7 and BT747, and when they are co-cultured with this NK92 with CD16 DNA NK92 with CD16-CAR and 41BB intercellular domain, and they also use this commercial NK92 modified, that is NK92 CD713B, which is act as a control. And here what they are able to see is that the cell lysis with this NK92 41BB is significantly higher compared to the other conditions, and it increases with increase in the effector to target ratio. And similar results they are able to see for this BT747 also, which is also a HER2 positive cells, and these results are done in the presence of antibody, that is Trastuzumab at 10 microgram per ml, and co-culture time is 8 hours. So from both, and they have not shown the results for HER2 negative cells, but they have mentioned in the text that they do not see any significant difference in the HER2 negative cells. So considering this, it seems like that the NK92 mediated anti-tumor response in the form of apoptosis against the target cells and which is facilitated by ADCC, it is antibody dependent cellular cytotoxicity. So that's all. This is what they find out in terms of the mechanisms, that is, it triggers apoptosis, which is facilitated by antibody dependent cellular cytotoxicity. So because in the HER2 negative cells they do not see any enhanced lysis or enhanced apoptosis. This result, HER2 negative cells results are not in the main figure. It might be in the supplemental figures, but I have not checked the supplemental figures, and it is also not mentioned in the main text that these results are in the supplemental figures. So you can check the paper and see if the HER2 negative cells results are shown there or not. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.